started. So that's the difference there. It's not, I would say the major difference between embryonal carcinoma cells and embryonic stem cells are the source of isolation. Because you remember, if you inject embryonic stem cells to muscles of an animal, and then you're going to have a teratocarcinoma. And then if you culture the teratocarcinoma cells, you're going to get embryonal carcinoma cells, right? So I think they're very, very similar to each other, except that the source of isolation is different. And then the third cell type that can be considered pluripotent is epiblast stem cell. You will have to know what is epiblast. So I will skip that part, to later. So that's a cell type that, in terms of development, that occurs slightly later than in the cell mass. It's still an embryo, but it happened later. At a later stage, if you isolated the cells from the embryo, what you get is epiblast stem cells. I'm going to have a more detailed description in the following slide. And then the fourth cell type that can be considered pluripotent is embryonic germ stem cells. So what are those? Those are the stem cells that will give rise to sperm, oocyte, the egg cell. So they have an ancestor. So during embryogenesis, some cells has already been assigned to eventually become sperm and oocyte. So at the very, very beginning, before you even have all the organs, some cells have already been committed eventually to become the germ cells. So those embryonic germ stem cells are also pluripotent. So eventually, that's the so what I'm trying to say is, if you take those embryonic germ cells, you put into the tissue, you put into muscle of an animal, it's going to cause teratocarcinoma. Okay, that's one of the way we demonstrate if the cell is pluripotent, right? You have two ways. One's one's the formation of teratocarcinoma, the other is chimeral formation, right? If you inject that cell to the inner cell mass, to the blastocyst, eventually those cells will be mixed and contribute to an individual, and then in that individual you have cells of the original inner cell mass, but also the pluripotent stem cells you're putting there. Okay, that's two approaches we use to demonstrate the pluripotence. The embryonic germ stem cell is the ancestor of germ cells. And the last two types, one's called somatic cell nuclear transfer embryonic stem cells, the other is called induced pluripotent stem cells. Those two are about cell fate reprogramming. Those are not natural. It's by human manipulation. All the top four cell types is natural occurring. So you just take the natural source and put the cells in culture, you can get it. But the last two cell types, one is reprogrammed through somatic cell nuclear transfer. How you do that, we're going to describe that several lectures later. We're going to describe that. And lastly, you have the induced pluripotent stem cell, although we're going to discuss the details later. But probably you heard that you put some factors into any cell. Theoretically, you put into liver cells, heart cells, and those liver cells eventually can become pluripotent. That means they gain the ability to give an individual. They gain the ability to become many cell types down the road of differentiation. So you change the cell fate from a somatic cell to pluripotent. That's the last two cell types. It's artificial. You make it happen. Okay, so those we're going to discuss also. So this is a summary of the main cell types of pluripotency, or main pluripotent stem cells. So if you have questions, please feel free to interrupt and ask me. So I think we're not going to use the whole time, 1 hour today. So I don't know if everybody have taken developmental biology, or I don't know if your biology course covered this or not, but for the purpose of explaining of embryonic stem cell as well as the epiblast, I have to do an overview of early embryology. So this starts with animal mate, right? So the sperm will travel up the organ and then goes first to, it's the sperm goes into the uterus. That's not here. It's not here. The uterus should be around here, and then the sperm will travel up along the so-called fallopian tube or oviduct, right? That's the oviduct. The sperm will travel all the way up until it will meet the egg, the oocyte. So this is ovary, right? The oocyte will be released from the ovary, and the oocyte will travel downstream, moving toward the uterus, and they will meet, very close to the ovary. They will meet. The sperm will enter the egg. That's called fertilization. So now, because both are haploid, that means they have only one N. For human, that's 23 chromosomes, right? Now, after fertilization, they have 46 chromosomes. So that's fertilization. In mouse, we call this E0.5. Why we call this E0.5? Because that's how we do things. We put the animals together. Usually they are separated. Female and males, they are separated. So we put them together. So tomorrow we call it half a day because it's overnight. And tomorrow, because we expect that during nighttime they mated, and during daytime we call it half a day. So that's why we call it E0.5. That's how we count the time, So that's E0.5 fertilization occurred, and then you can consider the numbers here. E1.0, that's 1 day after mating. Okay, 1.5, 1.5 day after mating. So that's what happened on each time point after mating. At the beginning, the fertilized egg is also called a zygote, right? That's a zygote that have two pronuclei, one from sperm, the other from the oocyte itself. And then at E1.5, you start to have the first cell division. You create two cells out of the original fertilized egg. And then at E2, you have 4 cells. At E2.5, you have 8 cells. And then at E3.0, the 8 cells will get compacted. So at that point, you start to lose the ability to count, because the morphology will not tell you how many cells are there. It's very hard to count from that point on. But before that, you can count the cell number. You can by looking at that illustration, you can conclude that there is no change of the cell size. At the beginning, you have a big oocyte, but until the cells become the 8-cell compacted embryo, you don't see any increase of that ball. It's still the same size, but cell number keep on increasing. Okay, so at the beginning, it's just 1 cell, the oocyte, but at E3.0, you have 8 cells. So you have many cells in there, or because the so-called cleavage, that's cell division, but you call it cleavage because we don't have any net increase of cell of embryo size. Okay, but before that, all the cells are totipotent. That means all the cells can give rise to all the cell types. After that, you're going to have a major change of the development. Now you created the early blastocyst. That's the structure of early blastocyst. As you can see here, you have some cell here on one end of the embryo. That's why we call it inner cell mass. Okay, so inner cell mass there. And then if you isolated the cell, you get embryonic stem cells. But remember, at this point, that embryo is not implanted yet. It's not implanted. That means it's still free. It's in the cavity there. in the oviduct cavity. Or at this point, I should say it's uterus, because that's the oviduct, and then it's the mouse uterus is a hole. It's long. It's not like a human. It's long, so that's a horn. But it's free. It's in the cavity. But once it gets implanted, that also the time will pass, right? That about E4.0, that's 4 days after fertilization, and now it gets implanted to the uterus wall. And at that stage, the embryo is called an epiblast. It's no longer blastocyst, it's an epiblast. So if you isolated the cell, it's no longer in the cell mass. It's called epiblast. If you take the cell mass here, you put into culture, they can also become a pluripotent stem cell, but they're called epi stem cells, epiblast stem cells. It's no longer embryonic stem cells, okay? Although they're still in a developing embryo, but you assign different terms to them. So that's the difference between embryonic stem cell and epiblast-derived stem cell. So on the right side, it's just the human counterpart. The only difference there is timing. The human apparently will have to take more time to go through the process I just described. Okay, so we're not going to do humans study that by yourself. The only difference there is timing. So here we have a more detailed illustration of development, of early development, and I will repeat the terms again. So at the beginning, it's called a zygote, and that's called a polar body. Look at that protruding part is a polar body. That's the oocyte will divide, give you a polar body and an oocyte, right? So 2 pronucleus, and then it become 2 cell, and then 4 cell, 8 cell, and then at one stage, E3.0, this is called a compaction. The compaction, the cells now compare. You can compare the 8 cell stage and this stage. It's called compact morula for a reason, because the cells now squeeze against each other. The cell mass become compact. The early blastocyst is when the cells move to one end of the embryo, creating this mass called inner cell mass. But at late blastocyst, they start to implant. And then once implanted, we call this pre-streak embryo. That's around E4.5 and 5.5. You see that the embryo now starts to be very different from at the beginning. So see this rendition. It's a compaction happening here, and then this is called cavitation, because now you have a liquid-filled cavity here, and the cells start to differentiate. You see these red cells is because the inner cell mass now start to give rise to epiblast. So this is transition stage. You see some cells that start to differentiate. So you have epiblast at this stage, and after implantation, you have this structure. Let's study a little bit details of this E5.5 structure. The red cell there, the lower part of the embryo is called epiblast. Well, the green cell on top of the red cell, that is the trophectoderm. That's the trophectoderm. Eventually, this part will invade will react with the mother side, and then creating the embryo, the placenta of the embryo, because the placenta will have 2 parts. One's from the mom, one's from the baby. So this trophectoderm will give rise to the baby side of the placenta. Okay, but remember that this red cell will give you the epiblast, the epi stem cells. So we explained this already, how you establish embryonic stem cell culture. Basically, you take the blastocyst and then you put them in culture. So there are actually 2 ways to do this. One I guess some students were wondering, when we discussed this, wants to just put the whole blastocyst there. So make that embryonic stem cell to come out not by itself. That's called hatch. So the embryonic stem cell will hatch out of the blastocyst and then grow. So that's it. That's one way, very easy. You just put the blastocyst in culture. The other is you do a surgery. You kind of microsurgery. You pick those inner cell mass cells out and put those cells in culture without including, the outside. So that there are two ways to do it. But one thing I want to explain to you is you see those blue cells. The blue cells is called a feeder. That's how scientists initially did the experiment. You need to put some fibroblast there to provide some cell signaling so that the embryonic stem cells can proliferate. So that's called a feeder cells. It's usually mouse embryonic fibroblast. It's some fibroblast you created from an embryo. Okay, you put those cells as a feeder layer and then put the blastocyst on top. I had some personal experience doing this, but I cannot do this by myself. I usually ask help from others. So I mate the animal and then I count the time at exactly 3.5 days after mating. I will cut out the uterus. It's, as I said, it's a horn. It's V-shaped. So it's V-shaped because mouse can bear many babies. It's V-shaped. So I will cut that and I take it to a friend, and who will do something amazing. Who will use a mouse pipetter. They put a pipette in his mouth, and he will use a tube to flush out the blastocyst. Basically, use liquid to flush out the blastocyst, which are tiny, right? Which are tiny. Under microscope, he will just pick up the embryos in no time. In seconds, he'll pick up 20, 40 embryos. That's embryos, blastocyst. He will use the mouse pipette, boom, 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 pick them up, and then quickly, distribute those to individual wells of a multi-well plate. Very quickly. He did that within 1 minute. He said he's done. I was amazed. But eventually we'll culture up a lot of different embryonic stem cells. But each embryonic stem cell line is from one animal, right? From one embryo. On this Friday, the students will have a discussion of the history of the radiation of embryonic stem cell. You're going to learn that. But here, this picture is from Gil Martin's paper published in 1981. Gil Martin is one who made a major contribution to the isolation of embryonic stem cells, but she didn't win Nobel Prize. I don't know how they judge who to give the prize, but this is an illustration that she used. It's an improved the illustration. The original was black and white, and now it's put more art in there. So I will leave this to the discussion on Friday, but I just want to say something. Do you think the embryonic stem cell and the embryonal carcinoma cells are very different? I don't know. By morphology, they will appear slightly different. You will see the morphology of embryonic stem cell. I don't have a picture of embryonal carcinoma cells. By look, they appear different. But remember, embryonic carcinoma cell were cultured like I don't know, it's like I believe it's in the 60s, the 1960s. So that's 60 years ago. And then the culture condition and the supplement in the culture are very different from the culture condition we use to culture embryonic stem cells today. So maybe the morphology was because of the culture condition, the difference in culture condition. because I think if you check the criteria, you know, the embryonic carcinoma cells can also give rise to chimera and to another teratocarcinoma, right? Those are the criteria of pluripotent stem cells. So people say that the carcinoma cells have mutations, which is probably true because it's abnormal to begin with. You isolate it from an abnormal tissue, not the developing normal embryo. So there could be mutations, but will you be able to find some cell that doesn't have the mutation from the carcinoma cells? I think it is possible. Why I say this, I want you to think about this, is because once we have embryonic stem cells, people start to abandon embryonic carcinoma cell. Because at the beginning, people use embryonic carcinoma cell to study differentiation, because those cells can also differentiate toward many cell lineages. People have studied that, used that to study differentiation for more than 20 years. But later on, we have the normal, so-called normal embryonic stem cells, and people have abandoned the embryonic carcinoma cells. But if you continue to study, maybe you will prove that they're identical. Maybe just the source of isolation are different. Just that's some thinking from myself. Okay, so when you read, you can think about this question by yourself. Now I give you the look of the embryonic stem cell. You see those elongated shape, the cells of elongated shape. Those are the so-called mouse embryonic fibroblast. Their fibroblast serves as a feeder layer. Okay, so when the embryonic stem cell, when the in culture on top of these feeders, they form this very unique morphology. You see a very clear edge of the cell mass. It's a lot of cells there, right? It's not one cell. You have very clean edge. The cells feels like raised. It's a dome there. You know, you can feel maybe it's a dome there. It's not very flat, right? Kind of raised colony in there. And also, you can't distinguish the individual cell boundary. It's not because of the resolution of that image. It's just we'll have a hard time to distinguish the boundary of individual cells. So that's, you know, the characteristic of embryonic stem cell culture. So in the future, you know, this is how embryonic stem cell will look in culture. So originally, the cells are always cultured on a feeder of mouse embryonic stem cells. But later on, people find that you don't have to have that mouse embryonic stem cell. You can have some supplement. Like one supplement is called LIF, L-I-F. It's a strange name. Stands for leukemia inhibitory factor. It sounds like that factor will inhibit leukemia. Nothing to do with stem cell. That's just a growth factor. It's a protein, a growth factor that we supplement to stem cell culture, to embryonic stem cell culture. And nowadays, people also use so-called 2I supplement. The 2I is a combination of chemicals or inhibitors of two major cellular pathways. One is the mitogen-activated protein kinase pathway. The other is inhibitor of glycogen synthase kinase 3 pathway. So that suggests that maybe there's two kinases. One is MAP kinase, the other is GSK3 kinase. Maybe these two are related to differentiation. So if you have high activity of these two kinases, maybe the cells will differentiate. If you have inhibitors against these two pathways, maybe the cell will keep their pluripotency to maintain the status of embryonic stem cells. Okay, so I explained to you 3 ways to culture the embryonic stem cell. And now I want to explain to you how you can get human embryonic stem cell. In mouse, it's easy. You just flush out the blastocyst and you put them into culture. But apparently, that's not the same story that you are going to derive human embryonic stem cell. Nobody will allow you to flush out the embryo. It's just one embryo if it's already implanted. Nobody will allow you to do that. Not the parents, not the government. It's simply something you can't do to flush out like in mouse. So this is purely done. The isolation of human embryonic stem cell is done in the in vitro fertilization clinic. The couples have difficulty in conceiving a baby, so they go to the clinic, they donate eggs and sperm, and then you do an in vitro fertilization. Basically, you put the sperm into the egg mechanically or some other ways. I don't know the details. So now you can put the embryo in an incubator. It's going to go through the same process, not in uterus, but in an incubator, in some kind of culture vessel. And then it's going to go through the 2-cell, 4-cell, 8-cell morula stage, and then become blastocyst. But there's another major difference between isolation of human and mouse embryonic stem cells. So usually the scientists won't wait until the blastocyst stage. They'll put those cells of 2-cell stage, 4-cell stage, 8-cell stage, put them into culture on feeders. And then because the out layer of human embryo is quite thick, so they will use some method to dissolve the so-called zona pellucida. That's the out layer of the embryo. And then basically the difference here is you can take the cells from early stages, from several early stages. You don't have to wait until the blastocyst stage. So you can derive human embryonic stem cells this way. It's more flexible. But of course, this is restricted to your access to a facility and a lot of ethical regulations. So that's why the mouse embryonic stem cell was established in 1980, 1981. The human embryonic stem cells was first established in 2008. That's about 20 years later, 18 years later. But I would like to give you a comparison and also the criteria of pluripotent embryonic stem cells. We learned that they must have self-renewal ability, to maintain a normal karyotype. That means the chromosome numbers and the chromosome integrity should be maintained. And also they can self-renew forever. You can freeze them, take them out, and just make them to replicate. It's an unlimited source. You can use that source to create many tissue types. So that both should be fulfilled by both mouse and the human embryonic stem cells. That's one criteria. And then differentiate in vitro to give rise to many cell types. And as I said, you must demonstrate 3 germ layers, cells from 3 germ layers at least, because we have so many cell types. You cannot exhaust the possibilities, right? So as a minimum, you demonstrate that you can create cells from 3 germ layers. And that's why you need to at least demonstrate that ability, because you can't demonstrate the full potential. It's impossible. And then the third criterion is contribute in vivo to the formation of every cell in the embryo, including the germline, if you inject that into early embryo. That can be done for mouse embryonic stem cell, but not for human. You want to put human cells into the blastocyst of somebody's new baby? Not possible. It's not allowed. You're manipulating. You're trying to play God. It's something. It's just not happening. Okay. But one thing you can do is you can put the cells into muscles of animal to form a teratoma. So you can do that for both mouse and human embryonic stem cells. Oh, so here remember those. If you understand the content here, you don't have to try to memorize anything. Just when you ask you about the criteria, just remember the not memorizing. Just try to. We're going to repeat this kind of criteria again and again. So I don't want you to just to try to memorize something without understanding the real meaning of it. But here, something bad about human embryonic stem cells. So the mouse embryonic stem cell have a very standard isolation method. As I said, it's E3.5 blastocyst. You put them into culture, you do it, somebody else do it. We follow a very similar procedure. And usually you get the cells of quite similar, comparable quality. Maybe it's related to the isolation method, or maybe it's related to something else. Maybe the human development is more fragile, not like mouse, more robust. They can create many animals. We can create only one most times. So maybe the human embryo is more fragile. So the cells isolated actually display some abnormality, such as genetic instability. So basically, when you first isolate the human embryonic stem cell, you do a karyotyping. That's basically you count you study the integrity of chromosome by a very raw method. You count the ploidy, right? That's whether it is 1, 2, and 3, and whether it have an additional chromosome or missing chromosomes, things like that, called karyotyping. So they start to display from the very beginning some degree of aneuploidy. That means abnormal, missing or additional chromosomes. So from the very beginning, you could have that. And once you put the cells into culture, it will accumulate some additional changes. And over time, they will adapt to the culture condition. You're going to see even more abnormality. So we say that the human embryonic stem cell will display genetic instability. They're going to adapt to the in vitro culture condition by adding or deleting chromosomes, because the chromosomes have genes. They feel like some gene is not giving them advantage for them to grow in culture. They're going to delete those chromosomes, or some other genes will give them advantage. They will try to gain additional copies of certain chromosomes. So that's something you don't want it to happen, because if you're going to derive these cells to tissue for regeneration purpose, well, you don't know what that will cause. Maybe a cancer. That's possible, right? So that's some difference of human and mouse embryonic stem cell. But here, I want to give you even more difference about human and mouse embryonic stem cell, because they may represent different developmental stages. Remember my description of early development or the early embryo, okay? So here I want to tell you the culture condition difference. If you're culturing mouse embryonic stem cell, those cells will have a tight, rounded, multi-layer clump morphology. Remember that image I showed you? It's very tight. The colony looks round with a very clean edge, and then it looks, you have a dome there, right? Because the cell have multi-layer, so you see a kind of, you feel that the cell form a dome, okay? And in culture, you often supplement something like LIF or BMP. And for mouse embryonic germ cells, they also look very similar to mouse embryonic stem cell. Okay, so, but the look of the morphology are similar, the supplements are slightly different. The third pluripotent stem cell on this table is epiblast-derived stem cell. Again, mouse, remember, you capture the cell at a later stage, not the blastocyst stage, but epiblast cell stage. And now if you put those cells in culture, they're more flat. You don't see the dome-like structure, they're more flat, and the aggregate is loose. By that I mean maybe you can see the boundary of individual cells. If the aggregation is not so close to each other, and maybe you can see the boundary of individual cells, right? And also the cells are more flat. The colony are more flat. That's the look of epiblast-derived stem cell, and then you have to supplement fibroblast growth factor and activin. Those are growth factors to culture. And now pay attention to the next one. The next is human embryonic stem cell. They also show a flat, loose aggregate morphology. You also need to supplement FGF and activin. So here you can tell maybe the human embryonic stem cell is somewhat similar to the mouse epiblast stem cell. So that means the human embryonic stem cell somehow ended up at a later developmental stage. So I'll go to the isolation. During isolation, you take the cell from early developmental stages. It's earlier than blastocyst, right? But somehow, when you establish the culture, the cells like to be in the epiblast cell stage. Okay, so it's relatively later than the mouse embryonic stem cell. So here is the ground state epiblast. So basically, this is before implantation. Before implantation, the epiblast is kind of like the original inner cell mass. Okay, so that's that's called ground state epiblast. Basically, this ground state epiblast is the inner cell mass. I'm sorry, this term here is confusing. It's not actually epiblast. You take those cells, you got this nice looking embryonic stem cell, but if you allow that to get implanted, you have this cell mass, that's the epiblast, and now you see the edge is not as clean, the cell look flat, more flat, and you see the boundary of individual cells. Okay, that's the difference there. Why I bother to tell you all this? It's because, you know, scientists are trying to improve the quality of human embryonic stem cells. They're trying to somehow get cells to go back to the ground state. They're still developing ways so that you can maintain the integrity of those cells. It's proven that early cell types may be more stable. Their genetic stability is better. So you're trying to, to establish cell lines that are more like the mouse embryonic stem cells, not like the epiblast-derived stem cells. That's just something I want you to pay attention to because it's still a challenge in the research field. I'm glad today I'm going to be a little bit ahead of time for the very first time. So lastly, I will use 2 slides to explain to you what's the usefulness of embryonic stem cells in tissue regeneration. So what's the first step of using them? So I explained to you on this slide a method called hanging drop. So if you culture the embryonic stem cells in feeder layer or with leaf, so you see the morphology, they're just, the edge are often very, very clean. Not exactly in this image, but often the edge are very clean. And also you don't see the boundary of individual cells. So that's the culture of embryonic stem cells as a monolayer, but you can trypsinize the cell. That means you can lift the cells off the surface, and then you put them in a suspension culture. So look, it's very special. You put the cells as hanging drops. You put the drops to the lid of culture vessel. You culture drops of cells to the lid of the culture vessel, and then you invert that lid. So the droplet are in a hanging position. And by gravity, the cells in those droplets will form a ball. Okay, the ball is called embroid body. ambroid body. Again, a third adjective, ambroid. It looks like, this probably means looks like embryo, ambroid body. So this is called ambroid body. And then this ambroid body will differentiate it automatically. And then later on, you can take those ambroid bodies and put them into some surface again. And then you initiate some treatment, you give a different growth factor, you can induce those cells to different cell lineages. Make them to become heart cells, make them become neuron cells, all kinds of possibilities. So you change the extracellular cue so that they will go to the fate that you tell them to go. So on the last slide, I'm showing you some work of myself. Maybe this was something I did many years ago, maybe 15 years ago. I'm in the field of cardiac development and skeletal muscle development. On this image, I show you some cells I differentiated from embryonic stem cells, from mouse embryonic stem cells. So the heart are muscle cells. You can see the striation. The muscle have striation. That's the characteristic of skeletal muscle and heart muscle. You can clearly see the striation if you do an immune staining. And on the right side, those cells become heart cells. They have the very characteristic rhythmic beating phenotype. So you see the cells as a cluster, they contract. Even in cell culture, they do that. So I labeled the cell with a red fluorescence protein. That's a use of it, like I explained previously. I put an mCherry underneath the alpha-myosin heavy chain promoter. So this red fluorescence is under the control of a heart gene. And now the cells will get, the cells will express that red fluorescent protein, mCherry fluorescent protein. That's a construct I got from Dr. Rogerton. I did that. I can visualize the cells. I can visualize the cells when they differentiate. Okay, that's the end of today's lecture. You can leave if you don't have questions. Otherwise, you can stay to ask me. Thank you.